on microbial drug resistance, one of the largest threats to human health. In this video, you will learn about antibiotic resistance and the two basic ways that the bacteria become resistant, and how this resistance is transferred among natural communities.
Now let's talk a little bit more about mutations. Just like any other organism, when bacterial cells divide, they duplicate their genetic material. This material is composed of DNA. Portions of the long molecule can hold precise information on the activity and the development of the bacteria. These portions are what we call genes. No, not those kinds of genes. Each gene is composed of a sequence of 4 elements called nucleotides. 2 are always matched in pairs because they are complementary. DNA is copied very rapidly, so errors are very common. These errors are called mutations. Many of these mutations are useless and not harmful, but every now and then, one comes along that gives its organism an edge in survival. And for a bacterium, a mutation making it resistant to a certain antibiotic gives it quite the edge. As the non-resistant bacteria are killed off, which happens especially quickly in antibiotic-rich environments like hospitals, there is more room and resources for the resistant ones to thrive, passing along only the mutated genes that help them do so. The bacteria then multiply and multiply very fast. Up next is conjugation.
Genes are directly transferred from one bacterial cell to another. This requires direct contact between the cells via a bacterial pilus. This gene transfer can occur between cells from the same species as well as cells from 2 different species. A plasmid circle, otherwise known as a fertility factor, is required for conjugation. Strains of bacteria containing these plasmid circles are called F positive, whereas those without it are called F negative. F positive donor cells produce a pilus to connect with another recipient cell. To begin conjugation, the F factor is cut at a specific region called the origin of transfer by a relaxosome, a protein assembly, which then further associates with the strand to be transferred, known as tDNA strand. Then, accessory proteins of the relaxosome are released while a portion of the relaxosome, called the relaxase, remains attached to the tDNA. This relaxase is transferred to an exporter, which pumps the tDNA complex into the recipient cell. Once fully transferred, relaxase joins the ends to make a circular DNA molecule. It is then replicated to become double-stranded. Meanwhile, the same thing is going on in the donor cell. In the end, both cells wind up with a complete double-stranded copy of the F factor. This connection through the pilus is released, and each can go on to conjugate with other cells.
Next is transformation.
The second type of way that you can have a transfer of plasmids between 2 different bacterial cells is called transformation, where the bacterial cell that is still alive is able to gather residual DNA from outside of itself from a now dead cell. While this is a relatively rare form of genetic transfer, sometimes a cell dies and hence its nucleic acid, primarily its DNA, spill outwards as the cell decays or is destroyed. That DNA fragment can lie at least partially intact for long enough that another living cell can come by and accept the fragment and accept it into its own genome. This happens by a process called endocytosis, where the living cell is able to grab the old dead DNA and put it through its own cell membrane and eventually pass into the bacterial chromosome that's completed of the living cell. Then, the section of a genetic information, called a plasmid, can be inserted into the host's DNA itself and change its actual genome. The now changed nucleic acid can then perform different actions by creating different types of RNA, which will in turn produce different proteins of various functions. By making different possibilities during the translation and transcription, this is the basic hierarchy for all biology, going from DNA to RNA to proteins to then perform actions. The cell and its eventual descendants, since bacteria have cloned daughter cells thanks to binary fission, which we talked about earlier, the organism can use this to evolve out of the ashes of a phoenix from another dead cell and bring widespread change to the present.
Last is transduction.
In the last type of horizontal gene transfer, transduction, genes are transferred from one bacteria to another through bacterial viruses called bacteriophages. Bacteriophages are viruses that consist of DNA or RNA surrounded by a protein coat called a capsid. Bacteriophages, or simply phages, can only infect bacteria. In one method, the virus will incorporate a portion of its genome with the bacterial chromosome. In the second cycle, the virus injects its DNA or RNA into the bacterial host cell to replicate. Upon significant replication, the host cell lyses, bursting the cell. Genes transferred through transduction include toxins, enzymes for sugar fermentation, and drug resistance genes.
And this concludes our presentation on how bacteria become resistant to antibiotics. Today we learned about how resistance occurs by two different mechanisms: natural mutations, as well as the transfer of plasmids by conjugation, transformation, and transduction. Resistance to antibiotics is the biggest threat to human health. Therefore, we should do everything we can to prevent this. The ways we can contribute to antibiotic resistance is simply from the misuse of antibiotics. These include using outdated or weakened antibiotics, using antibiotics for the common cold and other inappropriate conditions, using antibiotics in animal feed, failure to complete the prescribed regimen, and using someone else's leftover prescription. Now let's do the best we can to prevent antibiotic resistance because, as we can see, this is a serious situation.